Hello everyone, I'm Robin, and in this video we are going to check out what reject strawberry wine looks like under a microscope and what the wild microbes are that are on some reject strawberries. If you didn't catch the last video that I posted, I am currently in the middle of making some reject strawberry brandy. And so the first step of making brandy is making wine. So I took some reject strawberries. These were strawberries that were about to go into the garbage, and boiled those to remove any wild microbes, and then fermented them using distiller's yeast. You can catch all of that and the rest of the recipe details in this video right here. I was also curious about what microbes could possibly be on these garbage strawberries or these reject strawberries. So before I boiled the strawberries, I took not even a jar full. I took a handful of these reject strawberries that had thawed and tossed that in a jar and just let it go. There's some good activity in there. There's not only bubbling, but there is also some growth on top. So at first, it appeared that there was some mold growing on top. However, that has since subsided, which is really interesting. And I got a microscope for Christmas, so we can actually look at what microbes are in that juice. So let's do that and compare it to the microbes that are in the wine, which should be Saccharomyces cerevisiae. Here we have the reject strawberry wine at 100 times magnification. So this is just one drop onto the slide. This is the reject strawberry wine at 400 times magnification. These all appear to be Saccharomyces cerevisiae. Here we have the strawberries that were allowed to wild ferment at 100 times magnification. Wow, there's just like so much going on. Let's look closer at this shrimp here. Okay, so the shrimp appears to be a colony of bacteria, would be my guess, because there's so little. We're at 400 times magnification. Oh, there's some yeast. Look at all those. Whoa. Wow. More yeast. Let's see what else we can find. So as I was moving through the slide, I was able to find a bunch of different colonies of both bacteria and yeast, and bacteria and yeast in colonies together. I'm not sure what type of bacteria this is. My guess is something like Acetobacter, but I honestly have no idea. The yeast I'm also not so sure about. They have a similar morphology to Saccharomyces cerevisiae, so perhaps they're another species of Saccharomyces that also has this circular cell shape. I did a lot of reading and research. My guess is that what I've been looking at that are not the bacteria, the yeast that I've been looking at under the microscope throughout the week are all different types of spoilage yeast that you find in wine. So spoilage yeast are yeast that are essentially not Saccharomyces cerevisiae that can cause off flavors. What was interesting is that earlier this week, I had looked at the juice that was wild fermenting, and the cells had a completely different shape. They were much more apiculate shape, so shaped like a lemon. I'm not even sure I'm using apiculate properly in a sentence, but they were shaped more like a lemon. That would lead me to believe that it was probably the species of spoilage yeast called Hansenea spora, or also known as Clocara. I'm not even sure that's how you pronounce either one of these. Anyways, this spoilage yeast grows during the initial stages of fermentation and then tends to die off as fermentation continues. And they are readily present in various types of fruits, especially grapes. Now, some other spoilage yeasts that it possibly could be are species of Zygosaccharomyces. They typically have a dumbbell shape, which doesn't really align with what I'm observing currently, but the Zygosaccharomyces does produce CO2, acetic acid, esters, and higher alcohols. And Zygosaccharomyces bailey is fructophilic yeast, meaning it really likes fructose. So you find it on a lot of fruits because fruits have fructose. These yeasts could also be species of Brettanomyces. So Brett has varied morphology. So it can have this circular ovoid morphology, or it could also have apiculate and elongated morphology or cell shape. I did have a little bit of growth on the top of these strawberries, so a little bit of a pellicle layer, and Brett produces a pellicle layer. But also, if you look closely, you can see inside the vacuoles what looks like empty space in the middle of these yeast cells. You can see these dancing bodies. Yeah, let's dive into that. When I was looking into Brett, various Brett species, EurekaBrewing.com had observed these dancing bodies within 2 different Brett species that they had looked at under the microscope. Now, what the heck are these dancing bodies? So these are most likely velutin granules. And velutin granules are the intracytoplasmic storage form of complex inorganic polyphosphate. Interestingly enough, the function of polyphosphate in eukaryotic cells is not well known, it's not defined, it's not understood, but it's believed to be the cell's form of adapting to stress. That's kind of a cop-out, I think, as well as osmoregulation. The colonies of the yeast are also really interesting because the colonies are standalone as well as coexisting, co-colonizing with the bacteria that are present. And the actual growth form of these colonies is kind of all over the place. These yeast colonies are both aggregative and snowflake growth forms. This colony in particular was really, really fascinating to me because of how tightly packed they are, and also how all of them had these dancing bodies, these volutin granules within their vacuoles. After a couple of days, I took another look at the juice from these wild fermenting strawberries underneath the microscope. And again, the yeast had seemed to change. They had much more of an elongated cell shape as well as apiculate. And I was no longer able to find the dancing bodies within the vacuoles. This is really interesting because depending on where I take samples will completely affect the microdiversity of what's on the slide. When I took a sample from a little higher up in the jar, I was just getting tons and tons and tons of bacteria with very little yeast. When I took a sample from a little bit lower in the jar, I was getting a lot more yeast. However, as I said, their cell shape had changed and there was no longer those dancing bodies present. And the colonies of the yeast had much more of a branched growth, more like a snowflake growth form. And that would actually lead me to believe that these yeast cells that we're looking at right now are species of Brett. These could potentially also just fall under the Saccharomyces umbrella. I have no idea. If you have any idea what I'm looking at, please leave a comment below and let me know what you think, because believe it or not, I have not taken a biology class since high school. And if you want to see more videos like this, make sure to like and subscribe. This is really interesting for me. And yeah, so of course, I wanted to share it with you guys. And please share this video with anyone you think would find this neat. But the I can do it. I can say this. Aggregative. There's both aggregate. This could also just be another Saccharomyces species. Why is microbiology so hard? Oh my gosh.